Let's begin by looking at the process of fertilization as well as some of the mechanisms behind axis specification and cleavage. The majority of frogs have external fertilization in which the male fertilizes the eggs as the female lays them. It's worth noting that there are a few species of frog that utilize internal fertilization, and in that case the eggs are fertilized inside the female's body before they are released. Now, frog eggs have a distinct polarity before fertilization, with the vegetal pole having a dense yolk and the animal pole having very little yolk. And in some species, the two poles actually have a distinct pigmentation. In this case, you can see that the vegetal pole is light in color, whereas the animal pole is darkly pigmented. Another important difference between the animal and vegetal poles is the uneven localization of both proteins and mRNAs. In amphibian eggs, fertilization can occur anywhere in the animal pole, and the point where the sperm enters the egg is important because it's what determines the dorsal ventral polarity of the egg. The side where the sperm enters is going to become the belly or ventral side of the embryo, while the side that is opposite to the entry of the sperm is what's going to give rise to the back or the dorsal side of the embryo. The establishment of the dorsal ventral axis in the frog involves the rotation of the cortical cytoplasm, which is going to require the microtubules of the oocyte to be reoriented so that they are parallel to each other, as shown here in part B. Now, this rearrangement of the microtubules is achieved by the microtubule organizing center or centriole of the sperm. As a consequence of the rearrangement of the microtubules, the outer cortical cytoplasm is able to move independently of the inner yolky cytoplasm. And in this way, the microtubules allow for the rotation of the cortical cytoplasm relative to the inner cytoplasm, and that's a rotation of about 30 degrees. The movement of the cortical cytoplasm sometimes exposes a region of the inner cytoplasm that appears gray, and so that's known as the gray crescent, and that is located directly opposite to the site of sperm entry. This gray crescent is important because this is the region where gastrulation will commence. So as I just mentioned, the region of the gray crescent marks the side that is opposite to the sperm entry. And even in species in which the gray crescent is not exposed, the cortical cytoplasm still rotates relative to the inner cytoplasm. And so that means that the side where gastrulation is going to begin is going to be opposite to the side of sperm entry. And so recall that this side where gastrulation starts, which is opposite to the side of sperm entry, becomes the dorsal side of the embryo or the back of the embryo. The pattern of cleavage in frogs is similar to that of the sea urchin in that it is radially symmetrical and holoblastic. However, recall that since the distribution of yolk in the amphibian oocyte is uneven, so that there is more yolk in the vegetal pole compared to the animal pole, the eggs divide via mesolecithal cleavage. And functionally, that means that the cells of the animal pole finish dividing slightly faster than the cells of the vegetal pole. As you can see from this image, the first division begins at the animal pole and slowly extends into the vegetal pole. And in species that expose a gray crescent, as shown here, the first cleavage, which is meridional, passes through both the animal and vegetal pole and divides the gray crescent evenly between the two resulting blastomeres. Before the first cleavage has passed through the vegetal pole, a second cleavage that is at right angles of the first one and also meridional begins. Unlike the first two cleavages, the third cleavage is equatorial. However, because of the presence of moderate amounts of yolk in the vegetal pole, the third cleavage furrow is not exactly at the equator, rather it's displaced toward the animal pole, as you can see here. As a result of that slight displacement in cleavage, the animal region becomes packed with numerous small cells that are known as micromeres, while the vegetal pole region contains relatively large yolk-laden cells that are known as macromeres. The stage at which the embryo has approximately 16 to 64 cells is known as the morula stage because the embryo looks like a mulberry. Now, once the blastocele forms, the embryo is then considered a blastula. Now, the blastocele is not merely a fluid-filled cavity, it actually plays important roles during development. One of these roles is to prevent the premature interaction between animal and vegetal pole cells. In amphibians, the cells of the animal pole will give rise to the ectodermal tissues, while those that are located under the blastocele will give rise to the mesodermal tissues, and those that are in the vegetal-most region will give rise to the endoderm. If the blastocele was not present and the animal cells were allowed to come in contact with the cells of the vegetal pole, the animal pole cells will become mesoderm instead of ectoderm. So the blastocele is important for keeping the ectoderm cells undifferentiated until during gastrulation cell movements lead to a rearrangement of these cells so that they are able to be placed in regions that will allow them to develop normally as ectoderm. As in other organisms like Drosophila, the mid-blastular transition in amphibians involves a decrease in the frequency of cell division, which results from the acquisition of gap phases. Additionally, recall that at this particular stage, the genome of the zygote begins to be expressed, and that is achieved via both chromatin modifications, but also the demethylation of promoters. Now, this switch to the zygotic gene expression, however, involves the binding of maternally inherited transcription factors to the promoters of zygotic genes. Finally, another important consequence of the mid-blastular transition is that the cells acquire the capacity to move. And this capacity is important given the extensive cell movements that need to take place during gastrulation.